Okay, right, so welcome back to this next video in which we are discussing pain and analgesic drugs. Okay, so in the previous video what we did was we looked at the mechanism of the local anesthetic drugs, okay, and we saw how they only stop neurons firing if those neurons are trying to fire action potentials too rapidly. Okay, now one thing that I didn't stress in the previous video, which I want to now stress in this video, is that local anesthetic drugs work equally well against all different types of voltage-gated sodium channel. So remember, when we were looking at the structure of voltage-gated sodium channels in more detail, we discussed that there were those 9 different alpha subunits, okay, that you could use in building a voltage-gated sodium channel. Local anesthetics work equally well against all of them. Okay, so their mechanism is applies for all those different types of voltage-gated sodium channels. Okay, right. So what we now want to turn our attention to in this video is the mechanism of action of the opiate drugs. Okay, so examples of opiates include morphine and also codeine. Okay, so firstly let me just clear up what the difference between an opiate and an opioid is. Then I want to explain what the endogenous opioid system has to do with pain. Okay, specifically we'll look at stress-induced analgesia and the gating theory of pain. Okay, and then we'll see how opiates work. And unfortunately, opiates don't just have one site of action at which they work. They affect loads of different sites along this endogenous opioid system for relieving pain. Okay, so we'll have a look at all those different sites at which opiates act. Okay, right, so firstly, what's the difference between an opiate and an opioid? Okay, so an opiate is a chemical definition. Okay, an opiate is a molecule that has a similar structure to morphine. Okay, so it's chemically based. The definition of an opiate is chemically based, whereas opioid is biologically based, the definition of an opioid. An opioid is a molecule which produces in the body a similar effect to morphine. Okay, so opiate is really a chemist's definition, this is chemistry, okay, and opioid is the biological definition of any molecule which has a similar effect to morphine, basically, on the body. Okay, now, there are a number of endogenous opioids, i.e. molecules that are found within the body which do a similar thing to morphine. Okay, so I just want to talk about the different classes of endogenous opioids now, and then we'll discuss stress-induced analgesia and which endogenous opioids are involved in producing this stress-induced analgesic effect. Okay, right, so endogenous opioids, there are 3 major classes of endogenous opioids, okay, and they are the beta endorphins. So let me just put these here. So there is the beta endorphins, which contains the main example, beta endorphin. Okay, so beta endorphins, there are other beta endorphins, but the main example of a beta endorphin is beta endorphin itself. Okay, so that's one category. The second category is the enkephalins, okay, and these are the ones that are going to be extremely important for us. Okay, so I'll tell you about the 2 different enkephalins then. So there is methionine enkephalin, which is just abbreviated as met-enkephalin, okay, and then the other enkephalin is leucine enkephalin, which is just abbreviated LEU enkephalin. Okay, right, so those 2 are going to be extremely important for us. These are the 2 that are going to be involved in this endogenous analgesic system that's going to be employed in stress-induced analgesia. Okay, and then the final main category of endogenous opioids is then also the dynorphins. Okay, so dynorphins, and there are multiple dynorphins underneath that category, but we're not going to be particularly interested in the dynorphins in this video. Okay, right. So, let's talk about what morphine and codeine are agonists for. So they bind to a specific receptor, basically. Okay, and morphine and codeine, the main action, the main way in which they achieve their action is by binding to a receptor known as the mu-opioid receptor. Okay. However, there are more opioid receptors than just the mu-opioid receptor. Okay, so the main three opioid receptors are the mu-opioid receptor, the delta opioid receptor, and also the kappa opioid receptor. And all three of these receptors are G-protein coupled receptors. Okay, so if I draw the membrane here, the receptor will have 7 membrane spanning alpha helices in this classical way. And it will have its amino terminus extracellularly and its C terminus intracellularly. So there are these 3 main G-protein coupled receptors for opioids, okay? And morphine and codeine, they achieve their actions by being agonists at specifically the mu-opioid receptor. Okay, now if we're discussing the endogenous opioids, the beta-endorphin endogenous opioids, they are agonists at the mu and delta opioid receptors. The enkephalins, again, they are agonists at the mu and delta opioid receptors, but the dynorphins are agonists at the kappa opioid receptors. Okay, so this is why the enkephalins are the ones that are going to be involved in the endogenous pain relief system, because they are capable at least of activating the mu opioid receptors like these exogenous opioids here. Okay, beta endorphins aren't going to be majorly involved in the endogenous analgesic system, even though they are agonists at mu-opioid receptors. Okay, right, so let's now discuss stress-induced analgesia. Okay, so we'll go over the page and discuss stress-induced analgesia. Okay, so stress-induced analgesia, which is often abbreviated to SIA for short, is the phenomenon that soldiers in life or death situations usually don't feel pain, even though they have horrendous injuries in some cases. Okay, instead they feel the pain once they've been removed from the front line, basically. Okay, so when you are in an incredibly stressful situation, in a life or death situation, pain loses its physiological purpose, really. Remember, what's the point of pain? Okay, well, let's talk about inflammatory pain. What was the point of inflammatory pain? Well, it was basically to get you to treat that area which was damaged in a different way to the way you would treat it normally. Okay, to be careful with it. Okay, so that you allow it the best opportunity to heal, basically. But if you're in a life or death situation, that no longer becomes your biggest worry. That healing a small portion of your body doesn't become your main worry. Your main worry is surviving. Okay, so then basically the physiological purpose of the pain becomes irrelevant, and instead what can happen is you can shut down the pain coming from these inflamed areas, and this is what's known as stress-induced analgesia. And the key example is soldiers in life or death situations don't feel the pain associated with their injuries until they're actually removed from the front line. Then suddenly they become aware of the pain of their injuries. Okay, right, and it's because this endogenous system is activated to basically shut off pain. Okay, so let's now talk about the endogenous analgesic system that involves opioids big time. Okay, and underlies stress-induced analgesia. So basically, this story starts with the periaqueductal gray. It's believed that the periaqueductal gray, which is a portion of the midbrain, is incredibly involved in the stress-induced analgesic response. In fact, it's thought that when you activate the periaqueductal gray, this induces the stress-induced analgesia. Okay, so let's start by looking at the anatomy of the periaqueductal gray. So I'll just remind you of where the midbrain is. So remember, if I draw this picture of the brainstem, okay, here is the spinal cord, here is the medulla here. Above the medulla you then have the pons, and then the midbrain is the section that's above the pons here. So these are the 3 brainstem structures that we've seen before. Okay, now we're going to be interested in the midbrain, which is here in red. Okay, so what I'm now going to do is I'm going to draw a new picture of the midbrain where we're looking down on it from above rather than looking at it from the left-hand side. Okay, and if you look down on it from above, it actually has a much more interesting structure than you'd think from looking at it from the side. Okay, when you look at it from above, it has a Mickey Mouse face structure, like so. Okay, so here you have the two cerebral peduncles here, okay, which are like the ears of Mickey Mouse, okay, and within the midbrain, a landmark structure is the periaqueductal grey, so I'll mark this on now. Okay, so this is the periaqueductal grey, this tube right in the centre. This is what's known as the cerebral aqueduct. Okay, and this flows, or moves rather, through the entire length of the midbrain. So if I mark this on this picture here, moving through the midbrain, you have a little tube that is known as the cerebral aqueduct, and this is full of cerebrospinal fluid, and really it's the continuation of the central canal that goes through the spinal cord. Okay, and what actually it does is it connects to a large store of cerebrospinal fluid above the midbrain, which is known as the third ventricle, and it also connects to a store of cerebrospinal fluid that's below it, down behind the pons of the medulla, which is the fourth ventricle, and then the fourth ventricle connects into the central canal, which goes down the spinal cord. Okay, so this is just a little tube that goes through the midbrain, which has cerebrospinal fluid in it. Okay, so then this surrounding structure, this portion of neural tissue that surrounds the cerebral aqueduct, which is now coloured in red, this is very distinctive. When you look at a slice of the midbrain, you can certainly pick this point out. Okay, and this is what's known as the periaqueductal grey matter. Okay, the periaqueductal grey matter. And for short, the periaqueductal grey is usually abbreviated down to PAG. P for peri, A for aqueductal, and then G for grey. So I'll underline that in red. Okay, so this portion is thought to be the start of stress-induced analgesia. Okay, the neurons in the periaqueductal grey, interestingly, can be activated by mu-opioid receptors. Okay, so the neurons in here, I might just draw a little neuron here. So here's a cell body of this neuron. Basically, these neurons have mu-opioid receptors on them, and in fact, one of the sites of action of exogenously applied opiates, such as morphine or codeine, is that it can activate these mu-opioid receptors on the cells of the periaqueductal grey. Okay, now, the cells of the periaqueductal grey, they themselves actually use as their neurotransmitter endogenous opioids. Specifically, they use enkephalins as their neurotransmitters. Okay, and they send projections out of the periaqueductal grey to two other important structures that are going to be involved in this stress-induced analgesia. So let's now discuss these next two structures that are going to be involved here. Okay, so the periaqueductal grey is going to send projections to two other areas that are important in this stress-induced analgesia. One is what's known as the locus coeruleus, okay, and the other is known as the nucleus raphe magnus. Okay, so I want to show you where these two structures are in the brainstem and then what they're going to do. Okay, right, so we'll start off with the locus coeruleus. The locus coeruleus, well, you have two loci coeruleus, really. Okay, you have a left locus coeruleus and a right locus coeruleus, and they are at the back of the pons at this sort of level here. So let's take a cross-section of the pons, okay, and have a look where the locus coeruleus is. Okay, so if you take a cross-section of the pons, it ends up looking like this. Okay, you have these extensions towards the back like so, okay, which are known as the middle cerebellar peduncles, okay, which I didn't show on this diagram, but which are most definitely there. And these are connections from the pons to the cerebellum, which will be sitting behind. Okay, so these are fibers going from the pons into the cerebellum. So these are the middle cerebellar peduncles here. Okay, and here is the front portion of the pons, so this is the portion at the front here. Okay, so this is the middle cerebellar peduncle, and that one that I've specifically pointed to is the left middle cerebellar peduncle, and of course you have the right middle cerebellar peduncle here. Okay, right. Now the two locus coeruleae are at this sort of position here. Okay, so these are the locus coeruleuses. Okay, one there and one there. So left locus coeruleus, right locus coeruleus. I'll colour them also in blue there. So basically, these neurons in the periaqueductal grey are going to send projections down to the neurons in the locus coeruleus. Okay, like so. And these neurons are then going to be releasing enkephalins. Okay, so metenkephalin and leucine enkephalin. Okay, and these neurons in the locus coeruleus then also have mu opioid receptors on their surface. Okay, so here is the cell body of a neuron in the locus coeruleus, and it will now also have mu opioid receptors on its surface. So in fact, opiates can not only activate the periaqueductal grey, but they can also activate the things that the periaqueductal grey is going to activate. Okay, and that's how these opiates act at all the different levels of this pathway. Okay, right, now let me show you the nucleus raphe magnus, and then I'll talk about what the locus coeruleus and the nucleus raphe magnus are going to do collectively. Okay, so the nucleus raphe magnus, you again have two of them, and they're at the top of the medulla here. So now let's take a cross-section of the medulla so that we can see the two nucleus raphe magnus is. Okay, so basically if you take a cross-section of the medulla, it looks kind of like this. Okay, these are the pyramids of the medulla right at the front. This is the inferior olive, and then these portions behind are the inferior cerebellar peduncles. So from the medulla you also have connections with the cerebellum here, which are also input connections, neurons going into the cerebellum. Okay, so these are the inferior cerebellar peduncles, and again you have a left inferior cerebellar peduncle and a right inferior cerebellar peduncle. Okay, right, then moving forwards, these portions here, right at the front of the medulla, these are the corticospinal tracts moving down from the brain. These are the tracts carrying neurons which are sending instructions on how alpha motor neurons should fire. Okay, so these neurons connect to alpha motor neurons and control the contraction of skeletal muscle cells. So these are really important in voluntary movement, extremely important in voluntary movement. Okay, one here and one here. Okay, so as the corticospinal tract goes through the medulla, they're known as the pyramids. You can either call these the corticospinal tracts or the pyramidal tracts or just the pyramids of the medulla. Okay, but they're effectively just a huge great bundle of white matter that moves on the ventral surface of the medulla. Okay, then behind the pyramids, in these positions here, you have structures known as the inferior olives. Okay, also sometimes just called the inferior library nuclei. Okay, so in vivid purple, these are the inferior olives. And now we are ready to then show where the nucleus raphe magnus is. So this is the inferior olive, and that's specifically, again, the right inferior olive here. Okay, so let's show where the nucleus raphe magnus is then. So they're quite near the centre here, and I've abandoned the colour code, so I'll try and get the turquoise pen back again. So here they are, one on each side. So here's the left nucleus raphe magnus, here's the right nucleus raphe magnus. Okay, and again, you're going to have projections coming down from the periaqueductal grey. So I might show another periaqueductal grey neuron here, projecting down into the nucleus raphe magnus, like so. Okay, and again, you're going to be releasing enkephalins onto these neurons in the nucleus raphe magnus, and those neurons in the nucleus raphe magnus will have mu opioid receptors on their surface. Okay, right. So now, basically, when you activate the periaqueductal grey, okay, and one way to activate the periaqueductal grey is that the periaqueductal grey neurons have mu opioid receptors on their surface, and if you activate those, that will activate the periaqueductal grey neurons. they will fire action potentials down to these neurons in the nucleus raphe magnus and the locus coeruleus. Okay, and now if you then activate these neurons in the nucleus raphe magnus and locus coeruleus, again through mu-opioid receptors, because the periaqueductal grey neurons are releasing enkephalins, which act on the mu-opioid receptors, then these neurons are going to send axons down the spinal cord, basically. So these are going to send descending projections down to the spinal cord. Now, what are they going to synapse on in the spinal cord? Well, they're going to synapse on interneurons in the spinal cord, which use, again, enkephalins. So enkephalins are showing up everywhere. So let's go back to the dorsal horn of the spinal cord, because that's where everything is going to occur. Okay, so I'm going to redraw out the dorsal horn of the spinal cord here. So remember, the dorsal horn could be split up into these 6 rexed laminae, but the only ones we're going to need are number 1 to number 5. Okay, and to put on the stuff that we know, let's put on the spinothalamic projection neurons. So here is a spinothalamic projection neuron in rexed laminae number 1, and we also know there are spinothalamic projection neurons in number 5. Okay, and remember, A delta fiber primary nociceptive afferents will be synapsing onto dendrites of spinothalamic projection neurons in rexed laminae number 1, as will C fiber type primary nociceptive afferents. In addition, A delta fibers will actually brave it all the way down into rexed laminae number 5 to synapse onto dendrites of the spinothalamic projection neurons in rexed laminae number 5. The C fibers won't make it that far, but it doesn't matter, because the dendrites of these spinothalamic projection neurons in rexed laminae number 5 actually go into rexed laminae number 2, which remember is called the substantia gelatinosa. And basically the C fiber type primary nociceptive afferents can synapse with those dendrites in the substantia gelatinosa. Okay, right, so let's add a little bit of a little bit of colour onto this picture. Okay, so here are the second order spinothalamic projection neurons in red here. Okay, here are our C fibers in turquoise here. Okay, and here are our A delta fibers in orange. Okay, now I want to add a new type of neuron onto this picture that I didn't previously add at all, okay, which are neurons sitting in the substantia gelatinosa, rexed laminae number 2, and these are going to be interneurons, okay, in green here, and this specifically is going to be a type of interneuron that has enkephalins, okay, so we can call this an enkephalin containing interneuron. So enkephalin containing interneuron, and basically it's going to send projections to many of these synapses between primary nociceptive afferents and the dendrites of the second order spinothalamic projection neurons. So here it is going to have little synapses coming onto the it's going to have axon terminals synapsing onto synapses, okay, and it's going to be releasing enkephalins into those synapses, and we'll have a look at what those enkephalins do in just a moment. Okay, now, basically, these enkephalin-containing interneurons are going to be activated by descending projections coming from the locus coeruleus and the nucleus raphe magnus. And by the way, we often abbreviate locus coeruleus down to LC and nucleus raphe magnus down to NRM. Okay, so descending projections from the locus coeruleus are going to come down and activate these interneurons here, and so are descending projections from the nucleus raphe magnus. Now, neurons in the locus coeruleus use the neurotransmitter noradrenaline, okay, which we'll just abbreviate down to NA, okay, whilst neurons in the nucleus raphe magnus use the neurotransmitter serotonin, okay, which is often abbreviated down to 5-HT because its other name, its more proper name, is 5-hydroxytryptamine. Okay, right. So nucleus raphe magnus uses this neurotransmitter. Locus ceruleus uses noradrenaline. Okay, but both of these neurotransmitters have the same effect, which is that they activate these enkephalin-containing interneurons in rexathymine number 2. Okay, and now what do these interneurons do? They release enkephalins onto these synapses between the primary nociceptive afferents and the dendritic spines of the second-order spinothalamic projection neurons. And let's now see what this does. Okay, so let's firstly draw the synapse between our primary nociceptive afferent and the dendritic spine of our second-order spinothalamic projection neuron. Okay, like so. So I'll color code this again. So here is the dendritic spine of the dendrite of one of our spinothalamic projection neurons. Okay, and this is the axon terminal of one of our primary nociceptive afferents, and it could be a C-fiber type primary nociceptive afferent or an A-delta type primary nociceptive afferent. Okay, and basically now we're going to add a third little terminal on here, which is the terminal of one of these enkephalin-containing interneurons here. And basically, these enkephalin-containing interneurons, when they've been activated by either the noradrenaline or the 5-hydroxytryptamine coming down from these two high-up brain structures, they're going to release enkephalins onto the neurons of this synapse. Okay. Now, what is the enkephalins going to do? Well, basically, both of these neurons, both the presynaptic neuron, which is the axon terminal of the primary nociceptive neuron, and the postsynaptic neuron, which is the dendritic spine of the spinothalamic projection neuron, both of them have mu-opioid receptors, drawn here as these 7 membrane-spanning alpha helice structures. Okay, and when the enkephalins activate these mu-opioid receptors, it has effects on the presynaptic axon terminal and the postsynaptic dendritic spine. Okay, so the effect on the presynaptic axon terminal is to cause less neurotransmitter to be released. Okay, so less neurotransmitter is going to be released. And the effect on the postsynaptic dendritic spine is to make it such that for a certain amount of neurotransmitter that's released onto this postsynaptic dendritic spine, the amount of excitatory postsynaptic current that you generate is going to be lower. Okay, so the mu-opioid receptors here are going to lower the excitatory postsynaptic current. So presynaptically, you reduce the amount of neurotransmitter. Postsynaptically, you reduce the excitatory postsynaptic current. That therefore weakens this synapse, basically. Okay, it means now that the chance that this an action potential coming up this presynaptic primary nociceptive afferent is actually going to induce an action potential in our secondary spinothalamic projection neuron is much, much lower. The effect is hugely reduced. This is effectively central desensitization. Okay, and this concept is what's known as the gate theory of pain, because effectively these enkephalin-containing interneurons are closing the gate. They're closing the ability of the primary nociceptive afferents to actually induce activity in the second-order spinothalamic projection neurons. Okay, so that's what people mean when they talk about the gate theory. Okay, so to go through this entire stress-induced analgesia process, what originally happens is when you are in a very stressful situation, such as life or death situation, what happens is that the periaqueductal grey gets activated, and that's the portion that isn't understood yet. Upstream of this, we don't really know what actually causes the activation of the periaqueductal grey. The key thing to note is that the periaqueductal grey does have mu-opioid receptors on its surface and therefore can be activated by either endogenous opioids or exogenous opioids. Okay, so you activate the periaqueductal grey. The periaqueductal grey neurons use enkephalins as their neurotransmitters. They have projections down to these two structures, the locus ceruleus and the nucleus raphe magnus, and I've given up trying to say the plurals of those. Okay, and these will get enkephalins released into them. Okay, the enkephalins will activate mu-opioid receptors on the neurons of the locus coeruleus and the nucleus raphe magnus. These will then activate, they will send projections down into the dorsal horn, where they'll release either noradrenaline in the case of locus coeruleus or serotonin in the case of the nucleus raphe magnus, and these neurotransmitters will then activate enkephalin-containing interneurons in rexidlamine number 2 of the dorsal horn. These neurons are now the ones that actually close the gate. They then release enkephalins onto the synapses between the primary afferent nociceptive neurons and the second-order spinothalamic projection neurons. And this has presynaptic effects and postsynaptic effects. It reduces the amount of neurotransmitter released by the presynaptic neuron, and it reduces the response of a certain amount of neurotransmitter in the postsynaptic neuron, and therefore effectively closes this gate, reduces the ability of the primary nociceptive afferents to have an effect on the second-order spinothalamic projection neurons. Okay? And that is why in extreme stressful situations you don't feel the pain, even though your primary nociceptive afferents are still sending action potentials all the time. Okay, right, so how do opiate drugs then now work? Well, they work at all these different levels where there are mu-opioid receptors. So, morphine will work by activating the mu-opioid receptors in the periaqueductal grey and therefore setting off the entire thing. It will also work by activating directly the mu-opioid receptors in the locus coeruleus and the nucleus raphe magnus, also increasing the activation of these descending projections down to the enkephalin-containing interneurons. Okay, it will then also increase the actual effect at the gate, basically. It will work directly on the mu-opioid receptors on the presynaptic neurons and the postsynaptic neurons here, and therefore it alone can close the gate without even needing the endogenous system. Okay, so not only does it activate the endogenous system at 2 points by activating the periaqueductal grey and the locus coeruleus and nucleus raphe magnus, but also it completely circumvents all of that. It closes the gate itself by acting on the mu-opioid receptors on the presynaptic primary nociceptive afferents and the postsynaptic second-order spinothalamic projection neurons, and stopping the efficacy of this synapse, basically. Okay, so that's how opiates have their incredible capacity to treat pain and are an incredibly powerful branch of analgesic drugs. Okay, right, so in the next video what we're going to turn our attention onto is the mechanisms underlying neuropathic pain, and we'll look at drugs that are used to treat neuropathic pain.